of Public Health, Veterinarians, and he was a formal General Secretary of Indian Pharmacological Society and former member of State Board of Wildlife Forest Department, which is run by West Bengal government, state government. Dr. Tapan Mandal was born on 19 May 1958. Okay, so quite senior guy. Did his graduation in the year of 1982 from Bidan Krishi Vishwavidyalaya in district Nadiad of West Bengal state, and he did his post graduation in 1984 from Ranchi Veterinary College, and again he did his PhD in his UG college in the year of 1991 from Bidan Krishi Vishwavidyalaya, which is there in Nadia district of West Bengal state. Actually, because his passion and love for teaching and research work, instead of joining in department, animal husbandry department, he joined with the West Bengal University of Animal and Fisheries Sciences as an assistant professor in the Department of Veterinary Pharmacology and Toxicology in the year of 1985. So later, doctor got promoted as an associate professor in the year of 1995. And later, because of his dedication, subject knowledge and leadership qualities, he has been promoted as a professor of Department of Veterinary Pharmacology and Toxicology in Calcutta, West Bengal Animal and Fisheries University. Doctor is actively engaged in teaching and research work with sincerity for last 25 years. He is actually a paper setter and chief examiner for various reputed universities across India at national level examinations like ASRB and NET for accommodations. His research experience for more than 35 years in large animal, small ruminant and laboratory animal, extensively engaged in the work of pharmacokinetics and pharmacodynamics studies of various molecules, including some of the molecule which Alembic has launched in the past. Actually, the first study with Alembic doctor has done for isometamerium chloride, the efficacy of isometamerium chloride in Trypanosoma evansii, infection in a large animal, which is funded by Alembic Pharmaceutical. And again, ceftriaxone sodium, the efficacy of ceftriaxone sodium and the excretion level and the concentration of drug in the udder for every 24 hour or every 12 hour, how it is getting excreted. He has studied in goat. So by that way, he has fixed the dose for ceftriaxone sodium, our flagship product Emcept. Also, Dr. Tapan Mandal has studied the efficacy of aspirin in goat. That is also a project funded by Alembic Pharmaceutical. So one more paper, that evaluation of hepatoprotective efficacy of different formulations in Vista rats, that is also conducted by Dr. Tapan Mandal. Actually, Dr. Tapan Mandal has published 80 research papers in national and international papers or the journals. He has also guided 27 MUSC student and 14 PhD student during his academic career. He has also organized one international conference, 4 national conference, more than 13 membership scientific societies. He has participated and organized such events. Actually, we are all very, very happy and privileged to have such a remarkable researcher and accommodation with us to enrich our scientific knowledge and guiding us today. And Dr. Tapan Mandal is going to discuss on the topic which is there, that various antibiotics or chemotherapeutics and the merits and demerits of these drugs. So he is going to elaborate further on each group of antibiotics. So I welcome all the participants, the veterinarians across the country once again, and I happily hand over the session to our professor, Dr. Tapan Mandal sir. Sir, thank you. Over to Dr. Tapan Mandal sir.
Thank you, Mr. Kaunaniti, the Vice President of Alembic Pharmaceutical Limited. Good afternoon, the participants, particularly my senior and junior professional brothers and sisters, students also, and the organizer, Dr. Santosh, Dr. Gangadhar, Mr. Rajesh Sharma of Alembic Pharmaceuticals Limited. At the outset, I am really thankful to you for giving me the opportunity to deliver a lecture on this particular topic in front of veterinary professionals. The title of the lecture I have designed in this way, that clinical use of antibacterials in veterinary practices and overview. I know that everyone who are in veterinary practice, they know the antibacterials or chemotherapeutic agents, antibiotics, etc., and they are using every day about 75% of the prescription in case of your veterinary practice that include your chemotherapeutic agent. So this is not the new things to apprise you, just to recall you about these antibacterials which are being used in our veterinary practice. So before discussing the thing, first of all, you must know about the chemotherapy. There are some terms which are being confused sometimes among ourselves also, that is your chemotherapy. So chemotherapy means that we know, particularly general public and other than medical or veterinary professionals, that chemotherapy means it is the term used in case of your cancer treatment. But this is not at all true. Actually, chemotherapy, this term was coined by the father of chemotherapy, Paul Ehrlich. And according to Paul Ehrlich, what is chemotherapy? To describe the use of complex organic molecules or synthetic molecules to kill the invading organisms without affecting the host cell. Very important thing. We are using one chemicals that may be from your organic source or inorganic source or synthetic product, whatever it may be, we are using to kill the organisms. The organisms, that is your living cells, and our cells, that is also living cells. So when we are using one chemicals in our body, so that particular chemicals, it will kill that particular cell or tissue, but it will not affect the animal or human tissue. So that is very important. That means selection of this chemotherapeutic agent. Apart from this chemotherapeutic agent means, so it will also arrest the unnatural growth of the tissue or cells. That means in case of your cancer. So cancer cell growth will be prevented or it will be killed. That's all. It is known as your chemotherapy. Now the agents which are using in this condition, that is known as your chemotherapeutic agents. So any agents which can be used to kill the invading organisms, that is known as your chemotherapeutic agent. On the other hand, we have to know some antibacterials. So the agents which will kill the bacteria only, that is known as antibacterials. Antimicrobials, another term, which will kill the microorganisms, which may include your bacteria, your parasite, your fungus, and etc. So that is your antimicrobial. There is another term, that is your antibiotics. So antibiotics, the source of these antibiotics would always be from the living source. That may be from your bacteria, from fungus, from yeast, actinomycete, like this. So living source is the important. So chemical, organic chemical, which will be obtained from the living organisms, and that will be detrimental to the microorganisms. That will be detrimental to the living organisms. That is known as your antibiotics. So all the antibiotics are antibacterials, chemotherapeutic agent, but all antibacterials are not your antibiotic. All the chemotherapeutic agents are not the antibiotic. So that we have to keep in our mind. So in case of your antibiotics, in case of your penicillin, cephalosporin, they are the example. On the other hand, antibacterial, which are not your antibiotic, that is your quinolone derivatives, that is your sulfonamide group of drugs. So they are chemotherapeutic agent or antibacterial agent or antimicrobial agent, but they are not antibiotics. So keeping in view, I have restricted my lecture only on the antibacterial drug because this chemotherapy is the vast subject, vast chapter. This is not possible to cover in one lecture. So antibacterial drugs we are going to discuss. So first of all, we have to discuss about the antibiotics. So these antibiotics, before development of antibiotics, we got in 1908 the discovery of sulfonamide. So sulfonamide is not the antibiotics, it is antibacterial or chemotherapeutic agent. So it was discovered in 1908 and clinical use of sulfonamide started in the year 1928-30. So from that time, the golden era of chemotherapy started. So after discovery and after clinical use of sulfonamide. But most important thing, the discovery of penicillin, which was observed in case of your 1928 by Alexander Fleming, and the clinical use of penicillin, that is the first antibiotic developed, started from the year 1940-42, that is Second World War. And the modern era of chemotherapy started after discovery of penicillin. So this sulfonamide and penicillin discovery are very, very important for our chemotherapy. Now, in case of your penicillin, when it was discovered, so that was the magic bullet at that time. So wherever there was infection, that was extensively used penicillin. Now, in case of your penicillin, that we know, the penicillins, nowadays we are getting different types of penicillins and they can be classified according to their activity. So number one, narrow spectrum beta lactamase sensitive penicillins. Narrow spectrum, it will be effective against only gram-positive organism, but beta lactamase sensitive penicillin, so that will be destroyed by the beta lactamase enzyme, which is very important. So that enzyme is being excreted, liberated by different microorganisms, kill the penicillin nucleus, that is beta lactam nucleus, and that will inactivate the penicillin. So this is the problem, and that they are creating the resistance problem. Now, nanospectrum beta lactamase sensitive penicillins include your penicillin G, which is the first penicillin discovered. So benzyl penicillin, it is known as benzyl penicillin, penicillin G, and it is sometimes called as crystalline penicillin. And you know the use of penicillin, so it is generally used in case of your gram-positive organisms infection. And I have mentioned here the dose in case of dog and cat, 20,000 international units per kg body weight, and it is to be administered 4 to 6 times daily. Similarly, in case of your cattle, the dose is same, 10 to 20,000 IU per kg body weight. Then under this, procaine penicillin. So procaine penicillin G along with your procaine molecule. Procaine means that is your local anesthetic, which is mixed with penicillin G, so that penicillin G half-life will be increased due to presence of procaine. So due to presence of procaine penicillin G, the penicillin half-life has been increased to 24 hours. So we can repeat penicillin at 24-hour interval. Whereas in case of benzyl penicillin, we have to repeat at 6 to 8 hour interval. So in case of veterinary practice, we should know, we should have some long-acting type of drugs always for the benefit of our farmers. So procaine penicillin is another important in case of penicillin. Similarly, there is the benzathine penicillin. This is another long-acting penicillin. So in case of your cattle, the dose is 10 to 60,000 IU per kg body weight intramuscularly, and it is to be administered at single interval. So benzathine penicillin, procaine penicillin, these are known as your long-acting penicillin, and benzyl penicillin, this is your short-acting penicillin. So that will come under this group. Then number 2, narrow spectrum beta lactamase resistant penicillin. Narrow spectrum, effective against only particular type of organisms, but they are resistant to beta lactamase. So beta lactamase enzymes which are being liberated by the organisms, so that will be resistant. That penicillin, so that penicillin will not be broken down by the beta lactamase enzyme. So in presence of beta lactamase enzyme, these penicillin can act. They are of isoxazolyl penicillin. Under isoxazolyl penicillin, cloxacillin is one of them. Then oxacillin is another. Then flucloxacillin. So different types of trial on flucloxacillin also, and including Alembic Pharmaceutical also. In one time, they tried to introduce flucloxacillin. I do not know whether they have this type of flucloxacillin preparation. And then dicloxacillin. So if we compare oxacillin, flucloxacillin, dicloxacillin, so their activity will not vary major. So their activity more or less limited to the particular organisms. But the molecules are different due to changes of their structure. So sometimes we are using dicloxacillin along with your ampicillin and amoxicillin combination, and that is being marketed by different pharmaceutical companies, including Alembic Pharmaceutical also. Non-isoxazolyl penicillin, that include your methicillin, napcillin, and the dose rate in case of dog, cats, it has been available 10 mg per kg body weight orally, or it can be given intramuscularly, but 4 times daily. Then number 3, broad spectrum penicillin. Under broad spectrum penicillin, amino penicillins. Under amino penicillins, ampicillin, amoxicillin, tetracycline are the examples. So in case of your ampicillin, so these are the doses we schedule, you know, and that is being extensively used in the field condition. Then amoxicillin, this is another molecule. Just there is some changes in the structure of ampicillin, and we are getting amoxicillin. If we compare the ampicillin and amoxicillin regarding absorption in the system, so more or less same, but amoxicillin absorption will be better, about 100% compared to ampicillin, 95% in our body. So that amoxicillin is also being used in our veterinary practice in different susceptible organisms infection. Amoxicillin nowadays is also available as depot preparation. So the action, amoxicillin action or frequency of administration of amoxicillin has been increased by some pharmaceutical company also, and the dose will be 15 mg per kg body weight, and it is to be given subcutaneous route and 2 days interval. Then ampicillin precursors, example is tetracycline. This is generally used in case of dog and cat, not in case of your cattle. 10 to 40 mg per kg body weight, orally 2 to 3 times daily. There are other compound precursors, vacampicillin, divampicillin, talampicillin. So what is happening? So they will, if we administer these precursors, so in the body, after metabolism, they will be converted to ampicillin. Therefore, they are known as ampicillin precursors. And number 4, that is your extended spectrum penicillin. Extended spectrum penicillin. So they have the special effect on some specific organisms, and therefore they are known as extended spectrum penicillin. That does not mean they are broad spectrum in nature. They are specifically effective in some organisms. Say for example, Proteus, Pseudomonas, Klebsiella. So there were no drugs actually before the discovery of these penicillins. No drugs. They are gram-negative organisms, but they are specifically effective on these particular organisms, Proteus, Pseudomonas, Klebsiella, and therefore they are known as extended spectrum penicillin. Then it is of 2 types, carboxypenicillin. Example is carbenicillin. In case of dog, it is sometimes used at the dose rate of 55 mg to 110 mg per kg body weight, IP 3 times daily. Carbonyl indanyl salt that can also be used in case of UTI, dog, cat at this dose level. Tetracycline is another under this that can also be used in case of your dog and horse. Then uridopenicillin, the example is your meduloxylin, azulocillin. They are not being used in our veterinary practice. It is used in case of human practice. And number 6, that is your piperazine penicillin, piperacillin. Sometimes it is being used in case of dogs and cats at the dose rate of 25 to 50 milligram per kg intramuscularly 3 times daily. Then the term is potentiated penicillin. Potentiated penicillin. So penicillins will be activated by using some molecule. So example is amoxicillin, clavulanic acid combination. So if we use clavulanic acid along with amoxicillin, the amoxicillin activity will be increased. So tetracycline, clavulanic acid combination will increase the activity of tetracycline. So I am coming this combination just later on. There is another group under penicillin, that is your monobactam. They are beta-lactam antibiotics because they have the beta-lactam nucleus in their structure. Interesting, they are effective against gram-negative organisms. They are resistant to most beta-lactamase enzymes. That is very important. They are resistant to most beta-lactamase enzymes. Example is astrionam. This is not being used in our veterinary practice, only in case of human practice. They are being used extensively. Now, which I was discussing, that is your beta-lactamase inhibitors. So beta-lactamase inhibitors, we have seen that in case of penicillin, resistant penicillin, beta-lactamase resistant penicillin, and here this is your beta-lactamase inhibitors. Example, first compound, first molecule, that is your clavulanic acid. And by alteration the structure of clavulanic acid, we got sulbactam followed by tazobactam. So clavulanic acid, sulbactam, tazobactam, they are also antibiotics, belongs to beta-lactam groups, and they are the beta-lactamase inhibitor. What is that thing, and what is the difference between beta-lactamase resistant and your beta-lactamase inhibitor? Oxacillin, cloxacillin, dicloxacillin, and the difference with clavulanic acid, sulbactam, and tazobactam. In this condition, this clavulanic acid, they will inhibit the enzyme, beta-lactamase enzyme, which will be liberated by the microorganisms that will be destroyed by these molecules. Which molecules? Clavulanic acid, sulbactam, and tazobactam. So they will kill, they will destroy the beta-lactamase enzyme. So if beta-lactamase enzyme is destroyed, so say for example, it is being liberated by Staphylococcus aureus. So Staphylococcus aureus secrete beta-lactamase enzyme, and if we give clavulanic acid, so clavulanic acid will destroy the beta-lactamase enzyme, but the activity will be there, activity of the microorganism, that is your Staphylococcus aureus. So to kill the Staphylococcus aureus, we have to select one drug, one molecule, which will kill that organism. So here we have selected amoxicillin clavulanic acid combination. So amoxicillin clavulanic acid combination, if we use, what will happen? In, say for example, in case of your Staphylococcus aureus infection, so that enzyme which will be liberated by the organism, that will be destroyed by clavulanic acid, and the organisms will be destroyed by amoxicillin or ampicillin. So this is the scientific reason or basis or combination of clavulanic acid and other antibiotics. Clavulanic acid has no activity against your microorganisms. It cannot destroy, it cannot kill the microorganism. So combination, fixed drug combination is available in the market. So clavulanic acid, and if we compare the activity among these clavulanic acid, sulbactam and tazobactam, more or less same. There is no gross difference in the activity among these. So if we can combine with the clavulanic acid, that is better. There is no need of sulbactam. If you can combine with sulbactam, there is no difference, but only difference is the cost. And if you mix with tazobactam, the difference in your cost, nothing else. Then another antibiotics, development of antibiotics, cephalosporins. Regarding cephalosporin classification, on which basis we can classify? On the basis of generation. Nowadays, most of the veterinarians or veterinary doctors, all veterinarians, they are interested sometimes to know about the generation from the representatives of the pharmaceutical company. Which generation this antibiotics, which generation this molecule, which generation means if it is your 4th generation, 5th generation, 6th generation, that will be more effective according to their opinion. But it is not like that. Generation classification is not fixed. In case of cephalosporin, generation classification on the basis of spectrum of activity. Spectrum of activity. Cephalosporin is generally active when it was discovered against gram-positive organisms also. Like penicillin, cephalosporin was your narrow spectrum antibiotic. Now, due to development of synthetic product, development of different changes in the molecule of cephalosporin, we can get a variety, a number of cephalosporin derivatives nowadays. So they have been classified according to the generation basis, means at the spectrum of activity. So first generation means this group belong to those cephalosporin which are effective against gram-positive organisms. They are highly effective against gram-positive organisms and very, very less effective against gram-negative organisms. And they include cephalojolene, cephalexin, cephadroxil, cephaloridine, like this. So they are highly effective against gram-positive organisms and less effective against gram-negative organisms. That is your first generation. Second generation, they are effective against both gram-positive and gram-negative organisms. They are effective against both gram-positive and gram-negative organisms. We do not know what type of infection is there. So in case of mixed infection, we do not know. So in that condition, if we prescribe second generation, that will be more better. However, so they are effective against both gram-positive and gram-negative organisms, and they are second generation. Example is cefuroxime, cefaclor, cefamandol. So they are the second generation cephalosporin derivatives. And third generation, they are highly effective against gram-negative organisms and less effective against gram-positive organisms. Highly towards gram-negative and lesser effect on the gram-positive organisms. The examples are so many, and that are being used extensively nowadays, starting from your cefotaxime. So cefotaxime molecules was first come to the market in veterinary practice. Then other drugs are there. Now ceftiofur, one of the most important drugs. First time it was introduced, I think, by Alembic Pharmaceutical Company in the name of Except. Dose 1.1 to 2.2 milligram per kg body weight intramuscularly daily. Now interesting thing is this, in general, cephalosporin group of antibiotics, their biological half-life in animal system is very, very short. So it is to be repeated 3 times, 4 times, 2 times, depending on the molecule of cephalosporin. However, most of the cephalosporins are to be administered twice daily. And the dose generally 25 to 50 milligram per kg body weight. But due to discovery of ceftiofur and marketing of ceftiofur, so it is clear that this cephalosporin is very, very effective and the dose rate is very less compared to others and the duration of action is prolonged. So it can be repeated at 24-hour interval, ceftriaxone. So this ceftriaxone is very important drug nowadays, and we have to think about the use of these antibiotics, in which condition we have to give particular type of antibiotics or antibacterial agent. So ceftriaxone, it has been mentioned that it is effective in case of organism, susceptible organism of your respiratory tract, in case of your reproductive tract, so that are highly affected by ceftriaxone. So if you think to use ceftriaxone in other conditions, other susceptible organisms, so that may not be so much effective. So you have to think in your mind, or you have to develop yourself in such a manner so that in which condition we have to give this drug. So sometimes there is a failure of ceftriaxone in particular condition, and you will blame that ceftriaxone is not effective. So according to the instruction, you have to do, or you have to administer that particular ceftriaxone. Then ceftriaxone, it is being extensively used nowadays. Cattle 15 to 50, I told you in general 25 to 50 milligram per kg body weight. It is to be administered intramuscular, intravenous 2 times a day. So you can think how much amount is required and what will be the cost, whatever it may be, ceftriaxone is being extensively used. Nowadays we are using ceftriaxone along with your sulbactam, along with your tazobactam combination. So ceftriaxone, it is effective against, highly effective against gram-negative organisms. Another important thing I forgot to tell you about the cephalosporin group of drugs. They are more or less resistant to the beta-lactamage enzyme. Here that will be cephalosporinase enzyme. So that enzyme cannot easily destroy the cephalosporin molecule compared to penicillin. So they ourselves are resistant to this particular enzyme. In addition, we are using sometimes ceftriaxone along with sulbactam, ceftriaxone along with your tazobactam. That is your fixed drug combination, FDC. So that is another important. How much amount of ceftriaxone, how much amount of sulbactam or tazobactam is to be given. That is also fixed. So that is also being used in different conditions. Then cefoperazone, it is also being used, and cefoperazone, it is also mixed with your sulbactam or tazobactam nowadays. Then cefixime is another third generation cephalosporin that is used in case of particularly dog, high rise of temperature like that can be used. Then fourth generation, so they are the specific. They are effective against gram-negative organisms, no doubt. They will not be effective against your gram-positive organisms. So before that, I forgot one thing to tell you regarding ceftriaxone. So this ceftriaxone, this drug was given some trial in our laboratory. So one research scholar was there. Now he is the assistant professor in my department. In his PhD work, he was doing the work on ceftriaxone. So during his work, what happens? We studied the metabolites of the antibiotics, particularly in case of ceftriaxone. And we have identified ceftriaxone biological half-life very, very short. In case of mastitis, we have administered ceftriaxone and we have collected the milk sample at different time intervals and found that there is some concentration, and that concentration persists till 7 days. But that was not ceftriaxone. So we have identified that metabolite in the milk, and that was your ceftriaxone. And this ceftriaxone, it comes under your third generation cephalosporin, and ceftriaxone is being used in case of human beings. It was not being used in our veterinary practice. So first time we proved that ceftriaxone in the body is being metabolized to ceftriaxone, and that ceftriaxone is being excreted through milk. It can easily cross the blood-milk barrier and can persist in the mammary gland for a long time in case of ceftriaxone. And considering this, the Alembic Pharmaceutical Vice President at that time came to our university and discussed with our Vice Chancellor, and we have taken one project and that was successfully completed. That ceftriaxone, how far it is effective in case of your mastitis, now how many days it will persist in the milk. So that studies were conducted in our laboratory, and ceftriaxone first time it was introduced by this particular pharmaceutical company in the treatment of mastitis. So during our trial, during our experiment, we have seen that ceftriaxone, if it is used through intramuscular route, the erratic absorption will take place. Erratic absorption, sometimes it will be increased, highest concentration within 1 hour, sometimes it will take 5 hours, like that. So this route of administration should be avoided in case of ceftriaxone, but if it is used through intravenous route, it will be more effective and it can easily penetrate the blood-milk barrier and persist in the mammary gland for a long time. So therefore, it was marketed by that Alembic Pharmaceutical till date. Then fourth generation, we are coming. So there are cefepime, cefirom, and another compound, cefquinone. So that has come to the market also. Fourth generation, mainly effective against your gram-negative organisms. Specific organisms are also there. So this compound is another, or this molecule is another miracle molecule, because biological half-life with this molecule is also high. It can be repeated at 24-hour interval, and the dose rate is also very, very less. In case of cattle, 1 milligram per kg body weight intramuscularly and daily, and it has been seen that it is highly effective in case of mastitis caused by susceptible organisms. So it can be used. These are the mechanism of action. I am not going. Then antibiotics, that is your amino glycoside antibiotics. After discovery of cephalosporin, we got amino glycoside antibiotics. So amino glycoside antibiotics, there are so many derivatives, molecules. They have again been classified according to their spectrum of activity: narrow spectrum, broad spectrum, and extended spectrum. Narrow spectrum, they are effective against gram-negative organisms. Whenever it was discovered, first molecule, that was streptomycin, and it was effective against gram-negative organisms. And during discovery, at that time, streptomycin was the drug of choice in case of your typhoid fever, in case of your mycobacteria, tuberculosis. It was the drug of choice at that time, streptomycin, and it is effective against only gram-negative organisms, streptomycin. And this streptomycin combination is there, started from that time, that combination with your penicillin G. So penicillin, procaine or streptomycin, whatever, procaine or benzyl penicillin, whatever you need, but particularly procaine. So combination of streptomycin and procaine, which is known as streptopenicillin. So streptomycin, strepto, and streptopenicillin. So that is being marketed by different pharmaceutical company, including your Alembic Pharmaceutical also. So at that time, before cephalosporin development, so this combination, streptomycin and penicillin, was extensively used till date, these combination, because this will act as your broad spectrum. Penicillin effective against gram-positive, streptomycin effective against gram-negative organisms. So combination will act as broad spectrum, and they are known as streptopenicillin. So unique combination is there. Then broad spectrum aminoglycoside antibiotic, they will be effective against gram-negative organisms, to some extent gram-positive organisms. Neomycin, pharmacetine, kanamycin, paramomycin. So this neomycin, pharmacetine, kanamycin, they can be used as broad-spectrum aminoglycoside antibiotics. But this group of antibiotics, it has been seen that they are highly irritant. They are highly irritant. So neomycin, it was used previously through oral route, but neomycin or particularly aminoglycoside antibiotics, they are not absorbed from the GI tract because they are polycationic in nature. If we give through oral route, they cannot be absorbed, so they will remain in the GI tract for a long time, and therefore they are known as your gut-acting antibiotics. So for gut-acting antibiotics, these aminoglycoside antibiotics can be prescribed sometimes, but otherwise these drugs have to be administered through intramuscular or intravenous route. So neomycin nowadays is also available as injectable form, can be used through intramuscular route. Kanamycin nowadays it is also being used by our veterinarian in the treatment of sometimes mastitis and other conditions. And third, your extended spectrum aminoglycoside antibiotics, they are gentamicin, amikacin, tobramycin, sisomycin, erythromycin. So among these two are extensively used, that is your gentamicin and amikacin. So nowadays we are using gentamicin or we are using amikacin. They are effective against gram-negative major and less against your gram-positive organism also. So these are the dose of streptomycin, neomycin, kanamycin, gentamicin, amikacin. Important thing is this: in case of your gentamicin, you see. So dog and cat 5 mg per kg body intramuscularly it is to be administered, and cattle that also 2.5 to 5 mg per kg intramuscularly. But we have to administer twice daily. Our field veterinarian most of the time they are using 24-hour interval, that will not be so effective. So it is to be administered twice daily. Similarly, in case of your amikacin, if you see dog and cat 5 to 10 mg per kg body weight intramuscularly twice daily. And in case of cattle, the dose is higher, 10 mg per kg body weight intramuscularly, and it is to be given 3 times a day. Then we can consider about the tetracyclines group of antibiotics. So this is another old group of drugs, and there are so many molecules under these tetracyclines, and they can be classified according to their biological half-life, duration of action, short-acting, intermediate-acting, and long-acting. Short-acting, that is your Oxytetracycline, tetracycline, and chlortetracycline. Now, Oxytetracycline, this is very, very remarkable drug. Till date, this is being extensively used. Tetracycline was used, but nowadays it is not available in the market. Chlortetracycline, this drug cannot be absorbed from the GI tract, so it is restricted. Its efficacy in case of your GI tract infection, therefore it can also be considered as gut-acting antibiotic, chlortetracycline. So chlortetracycline absorption from the GI tract is very less. Therefore, they are known as gut-acting tetracyclines, and in case of gut infection, they can be used. But the use of chlortetracycline is only being restricted in case of poultry industry, not in our livestock, particularly your cattle and goat practices. Now, Oxytetracycline, that you know, I have nothing to say about Oxytetracycline, but Oxytetracycline, long-acting Oxytetracycline is also available, Oxytetracycline in LA. So, due to presence of particular carrier, particular vehicle, the Oxytetracycline's half-life will be increased in the system, and therefore we can use Oxytetracycline long-acting, and it can be repeated at 3 days or 5 days or 2 days interval. Oxytetracycline long-acting. The dose will be 20 mg per kg body weight, whereas only Oxytetracycline that is your 10 milligram per kg body weight. Intermediate acting tetracycline, dimeclocycline and metacycline, that is being used in case of your particularly human beings. Nowadays, there is some combination that I am coming. Long-acting tetracycline includes your doxycycline and minocycline. Doxycycline and minocycline. So these are long-acting because they will persist for a long time for their character. In case of your oxytetracycline, by mixing or vehicle, we have increased the half-life. But in case of doxycycline and minocycline, so there is no need of mixing any vehicle. So according to their lipophilic in nature, so they will persist in the body for a long time, and therefore it is known as long-acting tetracycline. Now, in case of your doxycycline, so doxycycline is used or was used in our veterinary practice, particularly in case of your dog. There is one term, off-label drug. Off-label drug. That means this drug is not recommended for use in case of dog. It is recommended for human use, and human formulation is available. So I can use sometimes in our animal practice, and we have seen that it is effective. So that's why it is known as off-label drug. Off-label drug, it was used. But nowadays, doxycycline can be extensively used in our veterinary practice. So doxycycline specific effect, that is in case of your leptospirosis and ehrlichiosis in dog. So in dog, this is the drug of choice, doxycycline. Without this, we cannot do anything. Previously, doxycycline was available as tablet form, so it can be used in case of dog, but it will take more time to recover from this condition, leptospirosis or ehrlichiosis. Now, due to marketing of doxycycline in the form of injection, first time I think by Alembic Pharmaceutical Company in the name of Doxycycline. So Doxycycline 100 milligram powder is available along with 10 ml distilled water, and it is to be mixed with the powder, and it is to be administered at the dose rate of 5 milligram per kg body weight intravenously, 12-hour interval. So 5 milligram per kg body weight we have to give at 12-hour interval for 7 to 14 days in this condition, that is leptospirosis and ehrlichiosis in dog, so that we can use, and that is very important drug nowadays, and we can do the treatment in case of your ehrlichiosis in dog. Now there is a combination for use in case of your poultry industry, that is your doxycycline and dimeclocycline. Sorry, doxycycline and neomycin. So combination of doxycycline, that is your tetracycline group of antibiotics, and neomycin that belongs to amino glycoside antibiotics. So neodox or like that. So this combination was not, I think, justified because doxycycline belongs to broad spectrum antibiotic, whereas neomycin, this is also broad spectrum but limited to your gram-negative. But most important thing is tetracycline group of antibiotics, they are bacteriostatic, while neomycin, that is your bactericidal. And as a rule, as a principle in your chemotherapy, we cannot use bacteriostatic drug and bactericidal drug simultaneously. Bactericidal drug means it will kill the bacteria. So neomycin will kill the particular organism. So when the organism will be killed by neomycin, so and in that condition, what doxycycline will do? It will inhibit the growth. But if there is no organism, so how it will act? So this combination, this type of combination is most unscientific. So those of oxytetracycline, doxycycline, minocycline, etc. Then another group, amphenicol. Under amphenicol, the first molecule, that is your chloramphenicol. So when it was discovered, chloramphenicol was the drug of choice in case of your typhoid fever, Salmonella typhi. It was highly effective at that time. But due to some side effect, particularly blood dysphasia, due to aplastic anemia, in case of human being, this chloramphenicol is not being used. But there is no such report of aplastic anemia in case of your animal treatment by chloramphenicol. So chloramphenicol can also be used in case of our veterinary practice. Chloramphenicol is effective against both gram-positive and gram-negative organisms. So it is truly your broad-spectrum antibiotic, like your tetracycline group of drug. Tetracyclines are truly, they are broad-spectrum, both gram-positive, gram-negative, apart from this chlamydia, mycoplasma. So here also amphenicol, they are also effective against this. So the dose I have mentioned here. Then chloramphenicol followed by thiamphenicol. In case of your thiamphenicol, just changing of the molecule. Structural changes are there, and due to changing, in case of thiamphenicol and fluorphenicol, it has been seen that they will not cause any aplastic anemia in case of your human beings. So in case of animals, also there is no chance of your aplastic anemia. So we can safely use your thiamphenicol and fluorphenicol. In case of your fluorphenicol, we have the advantage that biological half-life is very, very high, and therefore the drug is to be administered in case of susceptible organism infection at the rate of 20 mg per kg body weight intramuscularly at 2 days interval. Then we can think about the macrolides and lymphozones. Another group, that is your macrolides and lymphozones. This group of antibiotics, they are highly effective against gram-positive organisms. So gram-positive organisms, and in some specific conditions, specific diseases, they can be used. This macrolide and lymphozoide antibiotics. Macrolide antibiotics include the first member, that was your erythromycin, which was extensively used in case of human beings, and due to development of other derivatives under this, so this erythromycin nowadays is not being used. Then your tylosin is another important drug. It includes, it comes under your macrolide and lymphozoide derivatives. It is effective against gram-positive organisms. Particularly, it is the drug of choice in case of your mycoplasma infection. So mycoplasma in goat is a very problem. So it is the drug of choice. Mycoplasma is also a problem in case of your poultry industry. It is the drug of choice. So that is your tylosin. So tylosin can also be used in case of your cattle at the rate of 10 to 20 mg per kg intramuscularly, 12 hours or 24-hour interval. Pyramycin is there, polyandromycin and troliandromycin is also there. And then azithromycin. So these antibiotics, it is to some extent effective against gram-negative organisms. So sometimes it is known as your azithromycin as a broad spectrum. But this azithromycin nowadays is extensively used in case of human practice. Now azithromycin can also be used in our veterinary practice. There are some literature, there are some publication, it has been seen that azithromycin can be used in the treatment of babesiosis in dog. Babesiosis of dog treatment is very, very difficult. It is not easy to use like cattle, diminazine aceturate. It will not be successful every time, diminazine. Then there is imidazole nowadays available for your babesiosis in case of dog. Then another option is there. Recent literature is available for the treatment of babesiosis in dog. That is your use of buparvecum, which is used as thylaridiosis, in case of thylaridiosis treatment in cattle. So anti-thylaridal drug, buparvecon, can be used in the treatment of babesiosis, and it is to be given in the first day, followed by we have to give azithromycin for 5 days. 24-hour interval azithromycin is to be given single daily for 5 to 7 days, and there is chances of recovery from babesiosis in case of dog. So azithromycin can also be used in this situation. Besides, azithromycin can also be used nowadays as prokinetic drug, which will help particularly monogastric, particularly dog and cat, which will help in the empty of the stomach. So sometimes it is very difficult, overburden of the stomach or gastritis problem is there. So in that condition, empty. So prokinetic is the drug which will increase the motility of the, particularly your stomach and empty of the stomach. So in that condition, sometimes azithromycin is recommended. Doxycycline can also be used. Then your lincomycin, another very important drug. It is effective against gram-positive organism, no doubt, but it is the second line of treatment in case of your mycoplasma. If there is failure, if there is no drug in the market, tylosin, so in that condition, we can recommend your lincomycin in the treatment of mycoplasmosis. And in mycoplasma, the particularly cattle, the dose is 10 mg per kg body weight, twice daily. The drug is to be administered. So lincomycin application in case of dog is there, dermatitis, skin infection, pyoderma, soft tissue infection, which is a very and excellent drug that can be used, and that should be used through your parenteral route, because it has been seen that the absorption of lincomycin from the GI tract of the monogastric animal will be 30 to 40%. So very, very less absorption will take place. Therefore, this lincomycin should be recommended through intramuscular route in case of dog, at the dose of dog, 10 mg, 20 mg per kg body weight. Lincomycin can also be used in case of your anaplasmosis. So recent literature reveals that lincomycin is highly effective in case of anaplasmosis of cattle. So in case of anaplasmosis, there was the drug only Oxytetracycline for 8 days, 10 days, 15 days treatment. So instead of Oxytetracycline, you can think about the lincomycin. Then clindamycin, if we see, so lincomycin and clindamycin, there is no variation, more or less same, only there is some structural changes, and the efficacy of clindamycin is more compared to your lincomycin. The efficacy of clindamycin will be more compared to lincomycin, so that can also be used sometimes in case of your dog and cat. And I have seen that on literature, so clindamycin has been recommended in case of your babesiosis also. They have tried with clindamycin for a long period treatment. And then we are coming about the quinolone derivatives. So quinolone derivative, this is another old drug. These groups are not your antibiotics. They belong to synthetic compound. They can be synthesized in the laboratory, and they are used in different conditions. Nowadays, a number of molecules have been identified under your quinolones, and they have been classified according to the generation, according to the development. This classification is not like your cephalosporin. So according to development, so recent development, that means newer generation, before that second generation, before that first generation. So here also first generation. So first generation, first molecule, that nalidixic acid was extensively used once in the treatment of your GI tract infection, particularly in case of human beings, and in case of dog or cat. We have also used nalidixic acid orally. Then from nalidixic acid group, we have developed the molecule second generation, which include your enrotoxacin. So this enrofloxacin is another important drug. So enrofloxacin, if it is given to the body, so it will also be metabolized to your ciprofloxacin. So enrofloxacin is also effective against organisms, and your ciprofloxacin also effective against microorganisms. So enrofloxacin, we can think and define pharmaceutical company, including your alembic pharmaceutical in the name of enrocin, and it can be given at the dose rate of 5 mg per kg body weight, dog and cat, and cattle 5 mg per kg body weight intramuscularly. Nowadays, enrofloxacin has been used or marketed as long-acting enrofloxacin due to mixing of some vehicle, so that enrofloxacin will persist in the body for 2 days, 3 days like this, which is desirable in also veterinary practice. Difloxacin, another group which will come under your second generation, ciprofloxacin, orbifloxacin, danafloxacin. So danafloxacin is used nowadays in case of cattle at the rate of 1.25 mg per kg body weight intramuscularly or intravenously. Then margofloxacin, another compound. It is also being marketed by alembic pharmaceutical company. Injection model, I think, and it is effective against specifically pasteurellosis and mycoplasmosis. So Pasteurella infection, that is your HS infection, we have the drug sulfonamide, nalidixic group of drugs, but this margofloxacin is specifically effective against your hemorrhagic septicemia that can be used. Besides this, in case of your acute mastitis caused by E. coli and your mycoplasma. So mycoplasmal mastitis, E. coli mastitis can also be treated by margofloxacin in acute condition. The use of this margofloxacin is very interesting. It can be used as a single dose or a multiple dose. If we go for single dose, so we have to give that high dose, that is your 8 mg per kg body weight, and multiple dose we have to give at the dose rate of 2 mg per kg body weight, 3 to 5 days interval. %% solution of margofloxacin, the name of modal injection is available, marketed by your alembic. Then norfloxacin, ofloxacin, pfloxacin, but these drugs are not practically used, but there are so many work. Pharmacokinetic study has been done in case of pfloxacin, ofloxacin, but in the field condition we are not seeing these drugs marketed by any pharmaceutical company. And then the newer generation, that may be third generation, fourth generation, fifth generation like, but newer generation I have mentioned in this way, that include your gatifloxacin, sparfloxacin, moxifloxacin, primafloxacin. So these are also highly effective, and they called as new generation. Sparfloxacin, moxifloxacin, that has also been used, that has been studied by different pharmacology department in our country. And moxifloxacin nowadays is being also marketed by some pharmaceutical company in susceptible organisms. In disease condition, we can give moxifloxacin also. So with this, once again, I am thankful to the organizer, particularly Alembic Pharmaceuticals Limited, to give me the opportunity to deliver this lecture in very nutshell. Thank you. Thank you, all the participants.
Thank you very much, sir. I would like to invite Dr. Amit Singh and Naresh Kulkarni to take question answer session, sir.
Yeah.
Okay, sir. Actually, there are questions pertaining to this pharmacokinetic as well. So the first question is that tell us about the oral bioavailability of the antibiotics in case of large ruminant. There are always chances that it may degrade the ruminal microflora, and there are chances that it may kill the ruminal microflora as such. So which all molecules or the antibiotics will be betterly used as an oral antibiotic? The question is asked by Dr. Manikant.
Orally, it is used as oral administration.
Yes, sir. Bioavailability of the oral antibiotics he is talking about, sir. So Dr. Manikant is asking about the bioavailability of oral antibiotics.
Bioavailability of oral antibiotics. This is not the term. Bioavailability is the availability of the drug after oral, intramuscular, subcutaneous administration of the drug. So that we have to compare with the intravenous administration. So if we give intravenously, so that is 100% availability. But if we give through intramuscular, subcutaneous or orally, we cannot get sometimes 100%. There may be 40%, 60%, 90% like this, depending on the particular molecule. So in case of your aminoglycoside antibiotics, if we give oral load, so there will be no bioavailability, we can say very, very less bioavailability. I told you, so there will be no absorption because it is polycationic in nature. So it will remain as ionized form in the GI tract. Therefore, absorption will be very less. So that is applicable in case of your aminoglycoside antibiotics. And there are some antibiotics which can be used through oral route, but in case of ruminant particularly, so there is chances of inhibition of the growth of the rumen microorganism. So it is better to use through intramuscular or subcutaneous administration. But not only that, in case of your ceftriaxone, it is very interesting that if we give, not only ceftriaxone, other drugs are there. If we give through intramuscular or intravenous administration, the ceftriaxone will also be available in the GI tract. Transmembranal absorption will also take place. Transmembranal epithelial absorption will take place in this ceftriaxone. So this is very difficult to assess which drug will come to the GI tract, which drug will be absorbed or bioavailability will be 100%. So it will vary from drug to drug, antibiotic or antibacterial to antibiotic.
Yes, sir. Thank you, sir. Sir, there is one more question that what about the intrauterine antibiotics which are used? Is it safe or is it not safe as such?
Intrauterine injection or intrauterine administration, that is safe if we can use safely. Safely means by measuring some precaution. Say for example, if we give some irritant drug, so that will cause high irritation to this soft tissue. So that should be avoided. So considering this, we are to apply, we are to administer the drug through your intrauterine. So this is the practice. This is the practice of the veterinarian. So this ceftriaxone is available with me. So it is to be dissolved with water and you give through intrauterine. We have Doxytetracycline dissolved in water. Any other drugs are not available and we are giving intrauterine. So this practice should be avoided because there is one term that is your pharmaceutical dosage form. Pharmaceutical dosage form, that is very, very important. So pharmaceutic means pharmacist, means your M-Pharm and B-Pharm person, they will develop this technology. Which technology? They will develop the drug molecule in such a way, mixed with other molecules, so that the absorption or irritation or any other things, side effect, bad effect, that can be overcome, and that is to be used through your particular site, like your intrauterine. So that type of preparation we have to use instead of injectable form or oral form, that should not be used. Similarly, in case of your mastitis, so there is the tendency of our veterinarian to introduce intramammary. So intramammary ceftriaxone, dissolved in water or any other antibiotic, dissolved in water, you give. So it should be avoided. It may cause irritation, it cause further damage to the mammary gland. Instead of that, the formulation which is marketed by the particular pharmaceutical company, that should be used. Okay.
Okay, sir. So there is one more question that, in cases of dry cow therapy, although it is a very vast subject as such, in case of dry cow therapy, which is the best antibiotic to have a longer retention time in case of udder? So intramammary usage, intramammary usage, but which is the molecule which we can go for dry cow therapy?
No, there are so many compounds. We have so many antibacterials or antibiotics that can be used intramammary, but condition is they are to be prepared, they are to be made in such a way, in such a formula, so that they will not causing any harm to the soft tissue of the mammary gland. So that we can use your ceftriaxone, but ceftriaxone you have to use in such a formula. You cannot use the injectable form through intramammary route. That is avoided. Second important, we should not introduce the injectable form or other thing according to my opinion. So we are giving some intramammary infusion to the mammary gland. So we are going against the gravitational force. Gravitational force is there towards lower side, and we are giving the drug upper side. So how that drug will be dispersed in the mammary gland? So that is another question. How far it will be effective? Okay.
Okay, sir. Thank you, sir. So there is a question from Dr. Nilesh Sindhu, sir, that how we can define antibacterials and antimicrobials as such?
Antibacterials, it will be effective against bacteria. So bacteria against antibacterial. Antimicrobial means your microorganisms, bacteria, your fungus, like this. It will be broad, antimicrobial.
Yes, sir.
In case of your tetracycline, we can say your antimicrobial. It will affect gram-positive, gram-negative bacteria also. Then your mycoplasma, rickettsia, chlamydia. So it is your antimicrobial. And in case of your penicillin, this is your antibacterial, we can say. Limited.
Yes, sir.
Okay.
Yes, sir. Sir, is there any antibiotic which is contraindicated in cases of pregnancy as such? If so, which is this antibiotic?
Only in case of your aminoglycoside antibiotics, there is the contraindication of this in case of your pregnancy. So that will be, it will absorb the placental membrane and it will go to the fetus also, and it will affect the fetus, and there is toxicity in case of fetus. So therefore, your aminoglycoside antibiotics should be avoided in case of pregnancy.
Okay, sir. What is the correct dose in cases of enrofloxacillin that, some says that 5 mg, 2.5 mg. So how we can...
Actually, you see what is happening during your experimental situation. When the drug will come in the market, so they have given 2.5 to 5 milligram per kg on range. Now it has been seen in the condition, in the field condition, if you give 5 milligram per kg body weight, you cannot get any response. You have to increase the dose. So 5 to 10 milligram, that I cannot say that you have to use 10 milligram per kg body weight because I have no literature at this moment. But you are the practitioner. When you will practice, you will see that it is not so effective at the rate of 5 milligram. We have to increase the dose. Only things you have to see that how far it is toxic, how much it is toxic, what is the therapeutic index of that particular compound, and your LD50 of particular compound. Otherwise, if you increase the dose, so there is no chance of your toxicity at this level, twice or like that. In case of your penicillin, if we see that penicillin is the most safest drug, 150,000 international unit per kg body weight is the LD50 in case of your rat. So at this dose level, if we give, then 50% death will be there. So penicillin is very safe drug, but disadvantage in case of penicillin, that is your anaphylactic shock may develop sometimes. So in this way, you have to select, you have to judge actually which dose will be more appropriate in your area, in your animal. Sometimes you see the literature of the pharmaceutical company, dose as directed by the veterinary. It depends on you. You select how much you can give to get the desired response.
Okay, sir. So there is a one more question from Dr. Balaji Ambure, sir, that please elaborate on use of Oxytetracycline against Hemoprotozoal infections. As you were mentioning some Hemoprotozoal infections like Theileria, babesiosis and all. So this is the antibiotic which is commonly been used. So can you just elaborate the usage of this Oxytetracycline in case of
Oxytetracycline, it is actually in broad spectrum in nature. Till date, this is the old drug, and till date, it is the drug in different conditions we can use, and it is cheapest antibiotics. So in case of your hemoprotozoal infection, so in case of anaplasmosis, in case of filariasis, these drugs can be recommended, and the dose should be higher. In case of normal condition, normal disease condition, we are using at the dose rate of 10 milligram per kg body weight, but you have to increase the dose 10 to 20 milligram or 10 to 30 milligram per kg body weight. And it is to be given generally through your intravenous route, that will be better along with your saline. And that saline your D5, dextrose or normal saline, not with your Intose or any other things, Intalite like that. It should not be used along this type of electrolyte.
Okay, sir. Thank you, sir. There are so many questions, but because of the time constraint, we will give a full stop to the questions and answer session, sir. Over to Karnanidhi, sir.
Once again, sir, thank you for nice presentation to our professor, Dr. T.K. Mandal, because his expertise on pharmacology and toxicology. I am sure that many of the participants would have gained a lot of knowledge. Because of the time constraint, we are reducing the question answer session now. But still, whatever question you have raised, all questions are recorded. So all important questions we will get connected with Dr. Mandal later, and then we will try to answer it through mail. And all the participants will get a e-certificate for participating in this webinar. So that will be arranged by our PMT guy, technical guy, in the coming days. And I thank you once again for all of you for participating. I once again thanks Dr. T.K. Mandal sir for making a wonderful presentation. So thank you very much. Over to Dr. Santosh. You can make your closing remarks.
Thank you very much, sir. A warm and graceful evening to Honorable Tapan Mandal sir and all the participants. It is my pleasure to propose a vote of thanks speech and acknowledge the contribution of those who worked really hard to make this webinar successful. On the behalf of all the participants, Alembic Pharmaceutical Limited, myself Dr. Santosh Shinde, express my sincere gratitude to Tapan Mandal sir for such an informative presentation on clinical usage of antibacterial in veterinary practice, sir. Your presentation will definitely help the practicing field veterinarian to choose the antibiotic during day-to-day clinical practice, sir. In future also, we will take benefit of such an interesting and useful session that will upgrade our knowledge so we can serve the society. I would also like to thank Ritam Mandal, son of Dr. Tapan Mandal, sir, for helping making this webinar successful. Last but not least, I would like to thank you all the participating veterinarian, faculty and Alembic field staff for making this webinar successful. Thank you once again, Dr. Tapan Mandal sir and all the veterinarians.
Thank you, sir.
Thank you, sir. Thank you, sir.